Good evening. Hi. Great to have such an enthusiastic crowd tonight. Can you hear me, Francis? A little bit louder? Okay.
Hey folks, we're starting up.
Turn up a little bit. My name is Mark Lewis. I'm the host of Cafe Scientifique tonight, and to start, I'd like to acknowledge that we're on the Songhees, Sacramento, Esquimalt, Lekwungen territories, and we honor this with respect, and we also honor the Lekwungen and Wasanic peoples whose historical relationships continue with the land to this day. I am very happy to be able to introduce my good colleague, Steve Perlman, and I'm going to get my crib notes here. He is from Toronto, but he wants you to know that he hasn't been there since 1995. He is well-rounded. He did his undergrad at Toronto, majoring in zoology as well as French language and literature. He got stung by the subject of entomology, doing a master's at the University of Toronto, the Royal Ontario Museum on parasitic wasps. Not satisfied with regular-sized insects, he decided to drill down further and look at smaller parasitic nematodes that lived on insects, and this adventure earned him his PhD from the University of Arizona. As a typical academic, he bounced around at a couple of places as a postdoc before landing firm and square at the University of Victoria, from where he has not budged. He currently teaches a popular class in entomology and evolution, and for some reason it's often called Steveolution. So without further ado, let's welcome Steve.
Great, thanks so much. Can folks hear me? Yes? 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 No? Who can't hear me? Can you hear me now? Yes. Okay. All right. Oh my goodness, those lights. It's like, I just wanted to say...
Do you want me to turn it down a bit?
No, I don't know. Thank you so much for coming. I really appreciate it. It's a rainy day, and there's these friendly faces and other folks that are new friendly faces. So hopefully you'll humor me for 40 minutes, although it might be 6 hours, right? But I'll start with just apologies if I geek out or if there's just a little bit too much in terms of I forget myself of words that are a bit jargony. Maybe Doug, you'll make a sign or something, but I think it'll be all right. What else should I say? Café Scientifique. My daughter asked me if it meant it was going to be in French, and it was like, je ne pense pas. So it was, but let's start. So I'm a happy prof in the Department of Biology in the Faculty of Science at UVic. Not too far, of course, it's all part of our community, so come to campus. Everyone is so welcome there. There's so much fun stuff going on there. My talk today is called Microbial Manipulators, and the hardest part, of course, is that I don't have any slides. I have to do it only with my hands, but I have one prop just to start things off, and I need a helper.
Okay, I can.
Okay, and so, all right. Hold this up. I don't know if the folks in higher. Okay, can the folks in the back maybe see? Yes? All right, so I often show this image. This is an image from a lovely, lovely colleague, Richard Stouthammer, who just retired from the University of California at Riverside. And I often start by just asking folks, and no cheaters who already know the answer, like folks from my lab, but what is this? Thank you so I mean, usually when I teach an undergrad class, there's some silence, I need some cokes, but I heard a spaceship. Okay, anything else? It's not a spaceship.
Aubergine?
Aubergine, it is not an eggplant, no. It's not a nematode, no. It's not a paramecium. Lots of folks make that suggestion, and no, it's not a paramecium.
Nematode?
It's not a nematode, okay. All right, are you ready for the great reveal? All right, so maybe you can hold this back so I can point to it. So what is this? This is a picture of one of my favorite organisms, and an organism that I think you're going to fall in love with by the end of today. So this is actually an insect egg, okay, kind of amazing. And it's an insect egg that has been stained with a dye, and it's a dye that binds to DNA, and then it fluoresces, okay? So these glowing dots in the night sky, like dreams at night, all these halos of blue. So you can see a little bit of DNA, basically, that represents the nuclear DNA of the insect, but that's not interesting. I mean, it is interesting, but it's not the message of this talk. All of these halos of amazing lights, that's DNA. And what is that DNA? That's all bacteria. And so this is a very, very special thing, because this is a microbe, a bacterium. I also will use the word symbiont or endosymbiont. And when I speak about symbiont, I'm speaking about symbiosis, basically two organisms that are intimately associated with each other. Here's a bacterium that infects this insect host, and it's figured out the most amazing way to get into a host is that when the insect is born, it's already born with that infection already. So nice, so savvy, and so easy when you're a terrestrial, so a land-loving organism, and when you've got a microbe that is intracellular, living inside other cells. Isn't it so easy to just infect your host by being transmitted from mothers to their offspring, okay? And it turns out that this kind of strategy is very, very common for microbes, but especially for microbes that infect insects. It turns out that insects are very commonly infected with these types of microbes. Lots of different microbes have evolved that type of strategy to get passed on from mothers to their offspring so that the organisms are born infected with these microbes. And the specific microbe that I'm going to tell you about today is the most common one of all that does that. And there's going to be 3 words that I'm going to want you to remember throughout this talk, okay? And we're going to come to the first one, and it's hard, of course, because I don't have visuals, so we don't have the words, but the name of this amazing microbe, it's called Wolbachia, okay? So it's spelled W-O-L-B-A-C-H-I-A. It's named after Wolbach. Okay, so Wolbachia. Okay, so first thing I want everybody to do is to repeat after me. Wolbachia. Okay, just so that you remember it. Should we release Mark? Okay. Thank you so much. All right, and Wolbachia, if you can't remember it after today, I was trying to think of a nice way to remember it, and it's like, whoa, back off, okay? Okay? Will you remember this? Wolbachia? Okay. So Wolbachia, why is it magical? Well, insects are incredibly common. They're the most common, most abundant, and most diverse. It means that there are more species in terms of animals of insects than of anything else in the world. And it turns out that Wolbachia is really very good at infecting insects in this way. So it turns out that about 30% of all insect species and other relatives of insects like arthropods, like spiders, scorpions, isopods, and their relatives, and also some nematodes as well. But 30% of insects are infected with different strains of Wolbachia. And so what does that mean? That means that Wolbachia is the most abundant host-associated microbe on the planet. And so that's kind of amazing, and it's a little bit under the radar, right? We don't know about it. It's not famous like coronavirus or all sorts of other microbes, viruses, bacteria, microbial eukaryotes. So, but it's quite amazing. It's quite abundant, and it does a lot of really amazing things that I'll tell you just a little bit about. So a lot of diversity of Wolbachia strains, and basically every single Wolbachia in a different species does something sometimes just a little bit differently. So there's a lot of diversity, but each one might be a different strain with a different story to tell. There are a whole lot of microbes that have the same kind of biology that they get passed from mothers to their offspring, like Wolbachia, but Wolbachia is the most successful. And at the end of the day, we know still so little. Most of the infections, we don't even know which insects have got different microbes that they're infected with. And when we do, we don't even know what those strains of Wolbachia might be doing. But I will tell you what we do know, and of course I'm biased, but the things that Wolbachia is able to do, and how it's also gone from being a relatively obscure, basic research, kind of a cryptic, strange infection inside insect eggs, it actually has all sorts of really important applied consequences to studying Wolbachia. So I'll just take a pause there and say, is everyone all right? How's it going so far? Do folks remember the name of this microbe? Does anyone want to tell me what it is?
Wolbachia.
Yeah, okay, awesome. Great, thank you so much.
It's a little quiet back here.
It's a little quiet back there. Oh my goodness, it's so hard to...
Something. Move it down a bit.
Stay close.
How's that? Is that better? Should I start again? Well, that's a relief. All right. So what's special about Wolbachia, of course, what's special about Wolbachia is this strategy of being passed on from mothers to their offspring. So this is really interesting, right? So it means that this infection, it's intimately tied to its host. And so we could think that the evolutionary interests of a microbe that is passed on from a mother to the offspring are really, really intimately associated, because there's no other way, very, very rarely, that that microbe is going to be able to infect other things. And so it means that if that microbe infects an insect, a host, and that host produces more offspring, then of course there are going to be more microbes that are going to be passed along as well. And so that type of transmission from mothers to their offspring, it's very, very common that there are microbes then that provide a benefit to their hosts. And it's rare with Wolbachia, but there are many, many microbes that are passed on from mothers to their offspring that benefit their hosts so much that sometimes they become even essential, so that the host can't live or can't reproduce without them. And there are a few strains of Wolbachia that do that. I'm not going to tell you about them right now. They're relatively rare. I'm happy to chat about them if you're interested or if you want to ask in the Q&A. But those types of symbioses, they're called obligate symbioses. So of course the microbe can't live without the host. It's intracellular, it's inside a cell, it's lost the ability to even live independently. We can't grow it in a petri dish, we can't grow it on agar, we can't study it the way many microbes typically are studied. But we call them obligate symbioses because the hosts, in some cases, absolutely require them. Okay, and so that's very, very common in terms of microbes that are passed on from mothers to their offspring. And some strains of Wolbachia do that. Rare ones, I won't talk about those. What's much, much more common are microbes in terms of Wolbachia that are passed on from mothers to their offspring. They can't live independently, but the host without them. And so we might call that a facultative symbiosis in the sense that the insect, this insect for instance, it is capable of living without the Wolbachia infection, although the Wolbachia can't live without it. Okay? And that's what I'm going to be talking about for the rest of my talk in terms of the amazing things that these types of Wolbachia infections can do. So we're talking about how being passed on from mothers to offspring means that the evolutionary interests, the fitness of the microbe, of the symbiont, is really, really tightly linked to the fitness or the evolutionary interests of the host. But it turns out that they're not always perfectly aligned. And that's something that's really interesting. So what am I talking about? What do I mean? So Wolbachia, these types of microbes, they're only passed from mothers to their offspring. That means that anytime a Wolbachia ends up in a male insect, or a male spider, or a male host, that male is like an evolutionary dead end. It's not going to pass that microbe on because it's only passed from mothers to offspring inside the egg. And so that's interesting, but it means that the interests are not perfectly aligned. And so what does that mean? It means that there are some strains of Wolbachia, some strains of other microbes that are transmitted from mothers to their offspring that have evolved really amazing strategies. And this is the title of my talk, the microbial manipulators. And so what do I mean by these strategies? They've evolved interesting, sophisticated mechanisms to try to make there be more infected females. And of course that makes sense because these microbes are only passed through females, right? And so if somehow it can evolve a strategy to manipulate reproduction so that there are more infected females or fewer uninfected females, then of course more of those microbes will get passed along. And it turns out that those types of strategies, those manipulations, those reproductive manipulations are really diverse, really sophisticated, and there are a whole bunch of them. I'm only going to talk to you about one of them, which is the one that is the most common, the most abundant, the most widespread, and it's right under our noses, and it's also been the source of a lot of really interesting applied biology. So I'll just tell you about that one reproductive manipulation. And it's going to be the next thing that I'm going to try to get you to remember. All right, there's 3 words that I want you to remember. We've done Wolbachia, okay? And now we're going to do the next ones. Oh, bright, hot lights on. Keep going. And I'm getting my beverage. All right, so where are we?
The second word.
The second words, yeah. So what's this journey, right? I've told you about this really interesting bacterium, intracellular, only passed from mothers to their offspring inside the egg. And all kinds of interesting strategies that are associated with that. Some of those microbes end up being beneficial and essential for their hosts. And then there are others that are not essential for their hosts, but that have figured out and evolved strange tricks to make there be more infected females in the population. And I've teased you that I'm going to tell you one of those, the most common one. And here's what I want you to remember. Okay? Cytoplasmic incompatibility. Okay, you ready? How's that? So it's CI, 2 words, cytoplasmic. The cytoplasmic part, of course, is that they're passed in the cytoplasm, so in the egg, in that cytoplasm of the cell. Okay? And the incompatibility I'll tell you about in just a second. But let's step back, let's practice. Okay, so we'll start. What's the name of the microbe? Okay, and if you can't remember, right, it's like, whoa, back off, right? And what's the name of this reproductive manipulation?
Cytoplasmic incompatibility.
Okay, great. And if we wanted to shorten that, it would be shortened to? CI. CI, okay, this is great, not bad, huh? Okay. So you want to know what CI is and why it's amazing?
Yes.
Yes, okay. I'm so glad you said no. I would be crushed. So what's CI? So it turns out that this was something, basically a strange phenomenon that had been seen even in the 30s and the 40s in a bunch of different insects that in the 1930s, the 1940s, that researchers would, ecologists, they would basically bring insects. So, for example, mosquitoes, Culex pipiens, a nasty biter. They would take populations of Culex pipiens from different parts of the world, and they would bring them together, and they would try to get them to mate. And they noticed this very, very strange pattern called incompatibility. And it turned out that sometimes if you tried to mate insects from one species with insects of a population from another species, these very strange things would happen. The insects would lay eggs, but all the eggs would die. And so folks spent a lot of time trying to figure out what was that all about, how could you explain it? And it turned out that it's caused by the bacterium. It took a long time to figure out which bacterium it was. So in the 70s, folks were able to do some interesting experiments using antibiotics, for example, to try to get rid of the bacterial infection. And so that confirmed that it was bacterial. But then it was only in the 90s that folks really narrowed down that it was Wolbachia. And how does that work? What is that all about? So let's step back, because it's a little bit magical and subtle, this cytoplasmic incompatibility strategy. So how does it work? So remember, the microbe, the bacterium, it's only passed on from mothers to offspring. And it has evolved this magical strategy that it can do two amazing things. So here's the first thing that it does. It makes a poison, and it poisons the sperm of males that carry that infection. Okay? Kind of interesting, huh? And then the second thing that it does is that females that carry that infection, the microbe makes an antidote to that poison. Okay? So let's step back and let's try to figure out how on earth does that work? It's so nefarious. It's so mysterious. So let's start out with the case where you've got an insect. So a male doesn't have Wolbachia, doesn't have the infection, happy life, and doesn't have that microbe. So his sperm is not poisoned. And if he mates with a female that doesn't have Wolbachia, doesn't have the microbe, no problems. They mate, they're able to produce offspring, and those offspring are fine. If he mates with a female that has Wolbachia, it doesn't matter either, because his sperm doesn't have this poison, that this toxin that damages his sperm. All right, so we've got a few of the possible mating combinations figured out. Let's figure out what happens when that insect is infected, when that male insect is infected with a Wolbachia bacterium. Okay, so when that male is infected, now he's got a poisoned sperm. And if he mates with a female that doesn't have that microbe, that doesn't have Wolbachia, she doesn't have the antidote to that sperm. And what's interesting is that the microbe is not passed on through the dad. The sperm is, and the poisoned sperm is. she mates, she fertilizes her eggs, but there's a big problem because she doesn't have the antidote, because she doesn't have the microbe in her body, and all of her offspring, all of the eggs die. Kind of interesting, huh? Insidious, nefarious. But then what about the case where you've got the Wolbachia female that does, the female that does have the Wolbachia infection? Of course, she can mate with a male that has the poison sperm. She fertilizes her eggs with that poison sperm. But because she also has the microbe in her body, in her reproductive tissues, in her ovaries, she can also have that microbe in her eggs. It has the antidote to that poison sperm, and then she's able to reproduce, lay eggs. Those eggs hatch into happy baby mosquito larvae with great destinies. So kind of interesting, but let's step back and try to figure out, so why is that a useful strategy for the Wolbachia? Remember that the Wolbachia is only passed on from mothers to their offspring, right? And so what does that mean? That means that infected females, they're at a real advantage in a population, because the infected females, they can mate with infected males, and they can mate with uninfected males. So they can mate with any male in the population. And so they've got that advantage. And then what's doubly insidious is that their offspring, because they're passing on the bacteria to their offspring, all of their offspring, sons and daughters, are born with that infection. And so they're making not only more infected females that are passing on the microbe, they're making more infected males that are then going to be mating with the females in the population that don't have the bacteria, and then you're going to have fewer and fewer and fewer uninfected females in the population, which means you're going to have fewer and fewer population that don't have Wolbachia. Okay? Have folks got that? Sometimes it's a little bit so much easier with a slide. What do we need to know? We've got Wolbachia, right? Shall we say it one more time? Wolbachia. Okay. Cytoplasmic incompatibility.
Cytoplasmic incompatibility.
You guys are such great sports. CI?
CI.
Okay. And then the key is, right, is the sperm is poisoned, but there's the antidote in the females. Right? Kind of amazing. Who would have thought? I would never have thought this up myself. And so what's quite amazing is that if almost every individual in the population, if you can imagine that we are all mosquitoes in this room, all the men have got mosquitoes, have got bacteria. See the action of that incompatibility, because everybody's fine, because all the individuals have the poison, but they also all have the antidote. Kind of interesting, huh? Very, I keep using the word insidious, but it's also quite a successful strategy. And so it means that, so insects and their relatives, the most abundant animals on the planet, infected with so many of them infected with these different microbes, these Wolbachias, and most of these Wolbachias are causing this cytoplasmic incompatibility, and we don't even know about it, right? Because it's just under the radar. How am I doing for time?
About half an hour.
Right now. Yeah. Okay, this is great. All right, so I can even do this. All right, so is everyone doing all right? Did it seem like a little bit too much so far? The CI part, is everyone still with me? I think there are 2 exits, so I won't be offended. Now, of course, maybe not of course, I think what I'd love to tell you about is how CI, cytoplasmic incompatibility, is being used in amazing applied ways. Do folks want to hear about that? Yes. And then I'm also curious to hear what you think about it as well, because it's a very active area of insect control and disease control. All right. So there are basically two amazing ways that CI or CI microbes or CI Wolbachia or CI symbionts, however you want to call them, two amazing ways that they can be used to control insect pests, for example. I mean, lots of people care a lot about mosquitoes, understandably, because mosquitoes are basically the biggest killers of humans on the planet. And it's because of all of the diseases that they spread, all of the diseases that they vector, from malaria to dengue virus. All sorts of different mosquitoes cause all sorts of diseases in humans, in livestock. And of course, there are lots of insects that are also agricultural pests, economic scourges, lots of amazing insects which I adore. I'm an entomologist. But honestly, a lot of the applied interests in CI and in CI Wolbachia is to control insects that make our lives increase suffering in the world. So there are two ways that those are used. Okay, I'm going to tell you the first. I'll hope the second one for. So the first. So remember this idea, right? That if these males have got this microbe that makes poison sperm, and they mate with females that don't have the antidote, then all of those females' offspring will die. Okay? And so the first strategy is really interesting. It's not easy, but what people will do is that they will try to establish new infections in the lab. And so basically the idea is you take an insect that doesn't have a Wolbachia infection, or an insect that you want to give a new Wolbachia infection, and very, very carefully, very, very stressfully, not easy, you establish through embryonic microinjection, because remember, these things are intracellular, these things can't be, they live inside cells, they can't be grown independently. You want to try to establish a new infection. And you want to keep it in the lab, and you want to establish it in such a way that it's stable, so that your mosquito species that didn't have Wolbachia before is now transmitting stably this new Wolbachia infection. All right? And so strategy number one is that you then release into the wild only males. You got to be very, very careful. You don't want to release females, because if you release females with their new Wolbachia infection, they have the antidote, right? And they'll spread, and then your release strategy didn't work. But if you just release infected males, then the idea is those males will mate with females in the wild that don't have that Wolbachia infection, so they don't have the antidote, and then their offspring will die, and you'll achieve stable population suppression. And it's the kind of thing that you can stop whenever you like, or you can start it up again, and if it's an invasive mosquito, for example, you might not worry so much about having mosquito levels drop. If you're releasing males, males of course are not going to be the mosquito that bites, right? Because it's females that primarily bite. And so there are all sorts of potential benefits to that sort of strategy. Of course, you have to be very, very careful that you're not releasing any females by accident, and so it's quite labor intensive to make sure that you're breeding lots and lots of these mosquitoes, and there are all kinds of facilities that are doing this, and that you're making sure that you're only releasing males in the wild. But this is a strategy that is actually being used, and is actually being used with great success. And so one example, there's this subsidiary of Google called Debug. And what they have been able to do is they have bred new strains of mosquitoes that have a Wolbachia infection that hadn't happened before. So they basically took a Wolbachia from another insect, put it into these mosquitoes, and they're releasing them in Hawaii. And Hawaii, there are all kinds of invasive mosquitoes that are carrying all kinds of diseases, primarily species of malaria that attack birds, avian malaria. And so these mosquitoes are being released to be able to suppress these invasive mosquito populations, often in very inaccessible parts of Hawaii, that in an attempt to really save some of these endangered birds that are susceptible to avian malaria due to these invasive mosquitoes. So lots of excitement, lots of great promise from that strategy. You want to hear about the second one?
Yes.
Yes? Okay. How am I doing?
Oh, you're doing great.
I'm doing great. So the second strategy is really interesting. So remember, the first one is you're making a new infection in the lab. Remember, it's not genetic engineering, it's not genetic modifying, but what's interesting is you're making a new combination. And I'm curious to see what folks think about that, because it's a combination that hasn't been seen before, right? You're taking a strain of Wolbachia from one insect that causes CI, and you're putting it into another insect that hasn't seen that combination before. And that first strategy, right, you're releasing these males into the wild. Strategy number 2 is quite different. You're releasing now females. And you're like, what's that all about? So you're releasing females with this new strain of Wolbachia, and of course, because they've got the antidote to this poison, they're able to replace females in the population that don't have the Wolbachia, and they're just replacing them. And so you're starting to get mosquitoes now that didn't have Wolbachia becoming replaced by mosquitoes that had Wolbachia. So that's kind of interesting. But of course, one thing that's important, why would we want to do that, is if that new Wolbachia strain also has some sort of benefit along with it. And what's an amazing surprise in the field that I think, of course, I'm biased by the love of discovery and basic research, but this was a really big surprise that folks don't really understand yet how it works. But it turns out that some of these magical strains of Wolbachia, not only do they cause CI, but, and the drum roll, I mean, it's magical. When the first paper came out, people were stunned. Wolbachia suppresses RNA viruses. We don't know how, but the idea is there's only so much space in this insect, and they're competing for essential nutrients to build successful infections, and somehow there are strains of Wolbachia that make it that insects don't have RNA virus infections. So really interesting. Nobody knows really. That's one big frontier, is how they actually can do that. So what's an RNA virus that we would care about that Wolbachia seems to be able to suppress? I will tell you 3. Dengue virus, chikungunya virus, and Zika virus. And so what is magical, stunning, and this was first discovered by Luis Teixeira, totally unexpected, and now has been really well funded by the Gates Foundation. There's something called the World Mosquito Program, spearheaded by a researcher in Australia named Scott O'Neill. And they have managed to establish a new infection of Wolbachia CI in a species, Aedes aegypti, which is an invasive, the major vector of dengue virus and chikungunya and Zika virus. And they have released new Wolbachia-infected Aedes aegypti in many different parts of the world. And the first releases were in 2010, and so enough time has happened, and there have been some careful case-controlled studies where different sort of neighboring communities, particularly outside of Jakarta, is where the best data is, comparing whether or not the presence of these Aedes aegypti that have this new Wolbachia CI strain compared to ones that didn't. And guess what? The number of dengue virus has really plummeted. So it's this magical, very exciting frontier. And it's interesting to see where things go. We don't know how Wolbachia is able to suppress viruses. There's certainly benefits to these types of strategies in terms of mosquito control. They're very, very specific. So we're not talking about releasing chemicals, we're not talking about releasing pesticides, we're not talking about sort of off environmental effects in terms of other insects, and it's not transgenic. Two different combinations. I mean, I'm curious to see what people think about it. It's organisms that have never been brought together before. We don't know how or whether dengue virus is going to evolve and react. We don't know how Aedes aegypti with its new Wolbachia partner is going to evolve or react. But at least now in the short term, we see declines in terms of dengue virus infection. So that is quite interesting and quite exciting. How am I with time?
I think you're about 40 minutes.
I'm about 40 minutes. So, we're at a crossroads as we always are. So we have a bunch of different options, I think. Right? One is that I just stop-ish, right? I mean, one thing I was going to put out there is that I'm curious to know what people think about using this type of novel microbe combination as in insect control. This is something that I ask many of my students. In a sense, we're lucky in Victoria that we're not worrying about dengue, but there are many places in the world where there is not that luxury, and with changing climate and traffic and spread of all sorts of organisms, ticks, mosquitoes, everything in between, we might one day be asked whether or not we favor this type of control strategy in our backyard. And so that's something that I might have you percolate on. I'm also happy to say a little bit about the Wolbachia related research that we do in the humble, happy Perlman lab, but I will
Let the audience choose.
Carry on.
Okay.
Will there be time for questions, though?
Yeah, we
if
just
you
want to
carry
make sure
on?
there's enough time at the end for questions.
You tell me,
Okay, okay, so we'll say
5 minutes
5
for your
minutes.
lab.
5 minutes.
5 minutes, it'll be
Yeah.
less than 5 minutes for my lab. But the question is do folks want to hear, it's so hard because I think Maxine doesn't know if I'm going to put her on the spot, and she probably doesn't want to, and she's shaking her head. So there are two projects. I mean, there's lots of amazing projects in my lab. I'm so lucky, so blessed to be paid to study cool bugs. It's like a joy. But I'll just tell you about two things that maybe you might be interested in. So one very exciting frontier, I won't tell you too much about it, is that everyone assumes that Wolbachia is the one that causes all the CI in town. And of course, if you treat something with antibiotics, you're going to kill a whole lot of microbes. And a big discovery is that there are a whole bunch of other microbes that are also transmitted through the egg cytoplasm. And I think folks thought that it was always Wolbachia that was causing CI, but we're discovering new microbes that are able to do that as well. And so that's like a big frontier, is to try to understand Wolbachia infections and CI overestimated, or why do some microbes seem to do CI well or not well and in different ways? So that's one thing that we're studying that I'm pretty excited about in my lab. And then the other is, I didn't tell you that there are some rare strains of Wolbachia that don't cause CI. We don't know what they're doing, but they are essential. Can't get rid of them. And so in my lab, we've discovered one, and it is, of course, I'm biased, but it is a Wolbachia with the tiniest genome of all Wolbachias. It has only 500,000 base pairs of DNA, so almost nothing left. And so we're very excited to try to figure out, to use that as a clue to understanding what the Wolbachias that are absolutely essential, what they're actually doing, and why they're requiring. And Maxine in my lab is sort of spearheading that project, but I will not embarrass her too much, or folks in my lab. How's that? That was less than 5 minutes, right?
Thank you, Steve. Back to strangers and infection here. Let's open for questions, and I'll repeat your question when you ask it, so everyone can hear. I'll start, just while people are gathering themselves. What are the ethical considerations that people are generally coming up with here? I mean, I can see there's a whole slew of different things, and it must be front and foremost in some of the discussions. Can you talk about that?
Yeah, I mean, it's there's so much stuff going on in terms of different approaches for mosquito control, and so in terms of the microbe related, there are all sorts of jurisdictions that have tackled this sooner than later, but I would say that because nothing is being genetically engineered, that seems to put things in quite a different category. That we have a microbe that is already very, very common in an insect that is already very common. And so that seems to affect the logic in terms of regulatory bodies. But I mean, it's already since 2010 that there's been so much work in different jurisdictions. Australia was the first. Lots in South America right now, lots in Southeast Asia, a lot of Wolbachia releases that have been happening in the United States. And so there's always a lot of public consultation, and I think also because, I mean, it depends on the different flavors, but releasing male mosquitoes is a different flavor than releasing female mosquitoes, but for the most part, it seems like certainly folks have been thinking about these things, but there haven't been, everything's relative, but there haven't been the same kinds of controversies as some other approaches.
Thank you. Yeah, please.
How does this relate to the tick population that is kind of growing on an increase right now?
How does this relate to the tick population, which is growing and increasing?
Yeah, this is a great question. So ticks are amazing creatures, and it turns out that they're spreading a lot, and lots of tick diversity, and lots of diseases that ticks are vectoring as well. And so there is lots and lots of interest in the other microbes, the cryptic hidden microbes that infect ticks, and trying to figure out whether or not they might be useful or exploited isn't the right word, but taking advantage of them to try to use the same sorts of successes in mosquitoes as in ticks. Honestly, the field is not there at all, and I think it's just ticks are so hard. They're so hard to study in the lab, to breed, to maintain, they're much, much harder to work with than insects. But ticks have all sorts of interesting microbes, and there's a huge frontier of folks that are working on this.
Yeah, we'll start here. Yeah.
Sort of a comment rather than a question. I was so happy that you said bacterium. So many people have no idea what the singular form of bacteria is. It drives me nuts.
What would you have done if I'd said it terribly?
I would have approached you later and said,
Okay.
by the way.
Okay. Is there anything I didn't say?
The comment was you appreciated the use of the word bacterium, just so people at the back can hear that as well. Yeah, there was a question at the back.
Sort of two parts. How do you separate male and females? And then if you're releasing a bunch of males, how do you make sure that there aren't a few females that are going to kind of wreck the whole process?
How do you separate males and females and make sure you don't mix in the females with the males?
Yeah, it is. It's an amazing. I mean, it's not my. I'm not the one that's doing it. We don't work on mosquitoes in my lab, but it is an amazing, an interesting frontier as well. I've seen some talks where, I mean, Debug and Google, there are all sorts of amazing, very, very large scale factory level where they're trying to, so they have systems, for instance, where they have each different pupa. So basically the pupal stage before the mosquito emerges as an adult in complete metamorphosis, and based on size and certain features of the pupa, you can determine with fairly good accuracy whether or not it's going to be a male or a female. And you want to err on the conservative side, so you'll turf a few of the males as well. And so they go down this assembly line, and there are sensors, and they'll just so that's one example. For instance, some strategies are also to try to do a little bit of irradiation, and so you basically use a little bit like an X-ray so that you release, but they're slightly sterile, so that if by accident you release a few females, they won't reproduce. Or they won't successfully produce offspring. Some folks are even trying to use genetic engineering to genetically engineer so that only certain males are produced a certain way or females are produced a certain way. But people are spending a lot of time worrying about that and trying to make it work in terms of scale. So really interesting.
I think there was a question on the side, yeah.
You mentioned success with the dengue fever. Is there comparable success with malaria?
So the question was, you mentioned success with dengue fever. Is there comparable success with malaria?
Yeah, that's a great question. So the answer is no, and a couple of reasons why. So dengue and Zika and chikungunya are fairly easy for a couple of reasons, in the sense that there's one main vector, and that's Aedes aegypti, so an invasive, very worldwide mosquito. It's been successfully transfected with a stable Wolbachia infection. So that's one reason. The malaria is a problem for maybe three reasons. One is that Wolbachia strains, they're really good against suppressing RNA viruses. Malaria is not an RNA virus. So malaria is a microbial, a single-celled eukaryote, so like a protist. And so that part of the arm of Wolbachia's arsenal wouldn't work against it. The other strange thing, difficulty with malaria is that it's transmitted by many different species of Anopheles mosquito, and so that makes it harder. And then Anopheles seems to be relatively resistant to Wolbachia transfers. So the malaria stuff is far behind compared to the virus stuff.
Thanks. There's a question at the back. Go ahead, yeah.
As the science develops, do you think we'll reach a point where we can do other species, such as like actual animals and not insects? And do you think there are like dangers with humans being able to pick and choose what species lives and dies?
Do you want to repeat the question?
I would love it if you could repeat the question.
I'll try to get it right. So as the science progresses, and we are perhaps able to do similar things in other species, how might this progress, and what are the issues that come up?
Yes. Actually, maybe more insects are going to be adopted in terms of Wolbachia, but I don't think it's going to shift to things that aren't insects. And that's because for some reason Wolbachia and the strategy of Wolbachia infection and Wolbachia success and Wolbachia transmission is something that's very special to insects, to relatives of insects like ticks, so other arthropods, nematodes. So that type of strategy is not going to work in mammals, for example. So that's part one. And then part two, are there ethical issues with humans picking and choosing, doing anything And the answer is, of course. I mean, and it's interesting. I mean, it's a real discussion. I mean, it's not. I mean, I'm just one voice, but yeah, look at all of the mess we've made of things, and all the mess we've made of thinking that we can fix things in certain ways. And so it is interesting, sort of the hubris. I mean, the real excitement and discovery of being able to find Wolbachia and figure out that there's sperm that's poisoned and the poisons that do it, and all of the magic of being a scientist or a curious person to come to Herman's and in a basement and talk about these things. But yeah, I mean, it's we're not going to get ourselves out of this mess by constantly fixing new messes that we make and not fixing them well. But that's not what this talk or my life is about.
Yes.
I was just gonna ask you, you're talking about the issue of interference, and is there any benefit to having the non-Wolbachia infected mosquitoes in the population? You didn't actually say.
Is there any benefit to having the non-Wolbachia mosquitoes?
You know, in order to reduce the population, you're releasing the infection.
Yes.
Do the ones who don't have the infection, are they performing any kinds of, they have any kind of benefit?
Yeah, so this is also, I think that when these types of strategies were developed, and when I say, oh, you can release thousands of males and they will mate with females and there'll be population suppression, I think immediately there is this reaction, well, aren't mosquitoes good for the world? And of course, they most def are, and you know, they're important, play major roles in the food chain at different levels. And so don't misunderstand. I mean, I'm not advocating, and there's nothing about these strategies that means that there are not going to be any mosquitoes. Really, it just means localized suppression of especially invasive Aedes aegypti mosquitoes that weren't there before.
And just one last question, yeah, from the back.
I have a question about the bacteria that you were talking about, with 5,000 years. And I was thinking about with this specific one, and you also said that it's necessary for the insect to live. So I was thinking at this one point, what is the line between a semi-autonomous organelle and a separate organism that is an internal bacteria? Like, where do we draw the line, and how does this specific case help us understand it?
Yeah, that is, I think folks
Could
in my
you
lab,
repeat the question,
they
Steve?
love that question. So I'll repeat the question. So the question is, I mean, what I didn't speak about in terms of how important symbiosis is that we are all here because of symbioses. And so one example is the mitochondria. So mitochondria are specialized organelles in all of our cells, and they are incredibly old, and they are descended from a very, very ancient symbiosis where the ancestor of eukaryotes a long time back acquired a microbe as a symbiont that slowly lost almost all of its genome, all of its autonomy, lost a whole bunch of genes, sent a lot of genes into our nuclear genome, and now the mitochondria, even though there's still remnants of its bacterial history, is ours. And so I think the question is when do these Wolbachia symbionts and other symbionts, when do they cross that threshold to not being microbes and to being autonomous towards becoming organelles? And that is an awesome question, and there's it's something that drives the field a lot, and a lot of folks are interested in these microbes as windows into the early steps towards organelle and non-autonomous. In this case, there are still features, even though our Wolbachia is tiny, everything is relative. It's still much, much bigger than a mitochondrial genome. It has all sorts of things that it can encode and that it controls, for example, in terms of translation, replication, transcription, so it's still, it's not there yet. I'm not going to be here when it becomes that, but everything is on a gradient, and it's something that folks are really interested in.
Let's thank Steve Perlman once more. This concludes Cafe Scientifique for this year, and many thanks to Faculty of Science for sponsoring, and for the Mint for having us here, and for you, our audience.
Yes.
And for Mark for hosting us.
And we look forward very much to seeing you here in the fall. Have a great summer.